Okay.
Hello, my name is Bert Kosiski. I am the president of cureCADASIL, and I am here with two of my colleagues on the board of trustees of cureCADASIL. That is Benita Ball, who is a trustee, and Jane Gunther, who is our science officer and have been helping with this webinar. And we are very excited to have speaking today Joe Arboleda-Velasquez, who is a professor of ophthalmology at Harvard Medical School. And he's going to be talking on repurposing a pill therapy for treatment of CADASIL. And I must say, I think we have very good attendance today because we are all interested in finding treatments. Cure CADASIL is a nonprofit organization dedicated to helping patients, families, caregivers, and other supporters touched by CADASIL. We aim to unite patients and the medical community toward the common goal of treatments and ultimately a cure for this rare genetic disease by promoting communication, advocacy, research, and education. So Dr. Joe has worked on CADASIL for over 25 years from every angle, including working with families, biology, and experimental treatments. He has an MD and a PhD degree, the latter from Harvard Medical School. His laboratory is at the Mass Eye and Ear, a Massachusetts General affiliated hospital. Dr. Arboleda will be presenting data from his laboratory that introduces a new experimental therapeutic for CADASIL. The new therapy is a repurposed drug that has been tested for a different disease and was not approved but was found to be safe. He will show data showing how this promising therapy works in CADASIL models and his plan to bring this new therapy to CADASIL patients. If you have questions, know that the audience will be muted during this presentation, but we will take questions at the end. And if you have a question, you can type them into the Q&A menu. Questions will be answered either verbally at the end, or failing that, we'll type a message and send it to you privately. And also the questions should be repeated at the end of the webinar for everyone to hear. So without further ado, I am going to stop sharing my screen and invite Joe to share his. We'll see how this transition goes.
Can you see my presentation now?
Yes, excellent. And we
Thank
see you
you.
too.
Thank you. Welcome, everyone, to this webinar organized by cureCADASIL. I'm very grateful for the invitation, and it's just an honor for me to present this work. Yes, I have been working on CADASIL for quite some time. I was a medical student in Colombia, where I'm originally from, and CADASIL, you rarely hear the following expression. It's like CADASIL saves lives. Well, for me, it did, because it saved my academic career. I didn't know what to do with my life. I was bored in medical school, and I needed a project that I will find interesting. And that's when I found out about CADASIL, and it became my passion to work on CADASIL and learn about the biology. I became a scientist along the way, and the topic of my dissertation for my PhD at Harvard was on CADASIL, and this is how I began my laboratory. So yeah, CADASIL saved my academic life. You can say that, and it is an honor for me to present today this work, repurposing a pill therapy for the treatment of CADASIL. I understand many of you are people impacted by CADASIL or relatives. So my presentation is geared towards a lay audience, but happy to engage also via questions or later on with scientists interested in the work. First, so that I don't run out of time, I want to acknowledge that this work, I didn't do it by myself. I had a team in my laboratory. Key people include Dr. Rafael Posada and Said Arevalo, and then everybody else from the lab that have contributed. This work started with my great collaborator, Dr. Leo Kim, and you will see like a lot of his contributions along the way. I also want to acknowledge the families with CADASIL in Colombia. They, they have given me a lot of motivation over the years. I am more now in touch with families with CADASIL here in the United States, but they keep helping me from Colombia with their support, and they share samples and all of that. Many collaborators and funding this work has gone for a long time. So funding has come from all sources. I was maybe one of the first recipients of your CADASIL before you even remember, Bert. Thank you so much. Or you were not present at the time when this all began. So, but yeah, CADASIL has supported my research. American Heart Association, the NIH, Department of Defense, a lot of support goes into this. First, I want to do some disclosures. I am speaking from my academic post. As part of that work, I am listed as a co-inventor on a patent that got issued, and in that patent, we got claims for the use of RANX1 inhibitors. I'm going to tell you more about RANX1 for the treatment of CADASIL. That is kind of like what we are talking about here today. What goes in the pill that we are repurposing is a RANX1 inhibitor. And patents are very important to secure rights to inventions and then being able to advance them so that they become therapies and people can invest in that and then take them to the clinic. And as part of that effort, sometimes we academics also have to get involved and start companies to advance the work. That is also, I would say that is even more true for a rare disease like CADASIL, because a lot of people just don't care about CADASIL, or they never heard of. They would care if they knew about it, but they just don't. So yes, I co-founded a company. The name is X1 Biotech. The company licensed that technology and is advancing it towards the clinic. So those are my disclosures. Now, the work that I'm going to tell you about today has been going on for quite some time. And in collaboration with Dr. Leo Kim, the doctor is an ophthalmologist that I've been working with for, my God, more than 10 years. We have done a lot of work linking this RANX1 molecule to various conditions. And these are all the papers that we have published over the years, starting in 2017. Why am I showing you this? Because it's important that you know that, yes, I'm talking about a therapy and you're going to hear me very excited about it, but this is justified by the body of publications in various journals, and all of this has been peer reviewed. Starting, this work made it to the cover of the journal Diabetes back in 2017, and the last paper is in a very prestigious journal called Science Translational Medicine as recent as last year. In addition to that, when you judge the validity of a scientific discovery, it's important to know if, oh yeah, he has a lot of papers, but is everything coming from his own lab, or has other people gotten interested in this work and have validated it independently? And in fact, these are many papers that others have done independently of my work or that of my collaborator, and they have validated our results in terms of how this biology it's helpful for disease treatment. So it's a very well validated finding by work done in my laboratory, Dr. Leo Kim, and also independently. And there are papers now coming every other month on this, really. When Dr. Leo Kim and I were starting this, we're talking about, yeah, we're going to start a RANX1 revolution. It took some time to get it going, but it did get going. Now, what is this mysterious molecule that I've been talking about? It's called RANX1. Well, RANX1 is a transcription factor. I'm sure you guys have heard about genes. The genes are in our DNA and they make up proteins, sure. But it's not that these genes can make all proteins all the time at the same time. So who regulates that? Who regulates when genes are expressed and make proteins? Transcription factors do that. And one of them is called RANX1. So usually these transcription factors are in the nucleus, the part of the cell where the DNA is. And because they're in the nucleus, we call that being downstream. Downstream of what? Downstream of many growth factors. Growth factors are molecules that are outside the cell, and they regulate the function of the cell. And a lot of the time what they do is that they regulate the function of transcription factors. So RANX1 is a very important molecule and it has been around for decades doing all sorts of things. Part of what we discovered is how RANX1 plays a role in pathologies other than cancer. RANX1 was discovered in the context of cancer, but through the work of Dr. Leo Kim and I, we discovered that it also plays a role on other diseases that are not associated with cancer. Now, what I'm going to tell you today is this big discovery that happened recently. So yes, we have been working on RANX1 for quite some time. We just were not aware of the fact that RANX1 also play a role in CADASIL. And yes, I've been working on CADASIL for a long time, and in my laboratory, I have different projects. One of them related to RANX1, and then my CADASIL project. And these projects were like advancing in parallel, getting funded, losing funding, getting more funded, and publishing. And then at some point, it really hit me that these paths converge, that maybe everything was related, that maybe RANX1 also played a role in CADASIL, and that discovery was very recent. So first, I'm going to tell you how we identify this transcription factor called RANX1 as playing a key role in CADASIL. And for that, we use human tissue. Some of you might be involved in clinical studies and they want you to participate while you're alive. But you might have heard also that sometimes they say, well, if someone dies, they can donate the brain for research. And if you want to do that, we will be able to take the tissue and study it. That has happened, with many of my patients or the people that were my patients in Colombia, and now they die and they donate tissue. So we get to study that tissue. Participating in clinical research is very helpful for research, more so for a disease like CADASIL that has no treatment. So we were able to show that RANX1 plays a role in CADASIL by examining that tissue. And also we use animal models. You might have heard that there is this new trend towards using things other than animals. That is new. But for CADASIL, a lot of the data that we have involves mice, and we have what I think are great models of CADASIL in mice. I made some of those models as part of my PhD. I made mice and I put CADASIL mutations in them and generated and studied those models. That's part of like the biology that I did. And then I'm going to show you later some of this foundational work that Dr. Kim and I have been doing for quite some time. The models where we identify the role of RANX1 in eye disease and then in lung disease as well. But here I'm showing you an image. I'm sure if you have CADASIL or you have someone that has CADASIL, you are familiar with these images of the brain when you see these white spots that they call leukoencephalopathy, and that's not good. That means that there is a brain pathology and that neurons are dying. And some of them could be evidence of infarcts. I'm sure you heard about that. Yeah, patients, I say, always know more about the disease than we researchers do. So you are familiar with this. I just wanted to get everybody up to speed. CADASIL is the most common known cause of inherited cerebral small vessel disease. It affects our brain, in particular the vessels, more so the small vessels of the brain. And yes, it is a rare disease in terms of how many people actually get the strokes and the dementia, but the mutations that cause CADASIL could be a lot more frequent than we thought. It could be in like even like more than 3 or almost 4 per 1,000 individuals in the general population. And every time they look, they find that is more common than we thought. And now, again, just to get everybody up to speed, CADASIL is a vascular pathology. It affects the vessels. The vessels are the arteries that bring blood to the brain. And in CADASIL, they degenerate. So here is kind of like a cross section of the vessel. In a control, you see this white staining. That's the smooth muscle. The muscle is intact. In CADASIL, is not. Those cells are gone. And instead, I'm sure you heard about these deposits that accumulate, the granular osmophilic deposits. They are characteristic of CADASIL, and they kind of like take the space left by the smooth muscle cells that are gone. In a way, yeah, arteries in CADASIL patients like lack muscle. You can think about it that way in a simplistic way. Now, NOSH3, I'm sure you heard about that. That's the gene. That's the gene in CADASIL that gets mutated. And when it has a specific mutations, it leads to the pathology, in this case, the degeneration of the vessels. Now, these CADASIL variants in NOSH3 are the ones that are associated with the disease, and they usually involve, I'm sure you heard about that, like the cysteine residues or cysteine amino acids. If you have like those reports of the genetics from someone you know or yourself, it's always like a C, that stands for cysteine, followed by a number, followed by an R or another letter. Or sometimes it's a letter followed by a number, followed by a C. Cysteine amino acids are very important for CADASIL because they usually like to go in pairs. So in each of these repeats, part of the protein, you have 6, but in CADASIL, sometimes you have 5 or you have 7. And when there is one of these unpaired cysteines, the protein becomes very reactive and people think it could be toxic. There are many mutations discovered all over the world. Even in Colombia, we discovered some that were original to Colombia. And every time you talk about like these mutations, we always want to know, is the mutation like inactivating the protein, activating the protein too much, causing accumulation of the protein? What does it do? What does it do? It's always kind of like this mystery. And as a biologist, I spend a lot of time trying to figure that out. What is the biology? I'm sure you heard about this word mechanism. What is the mechanism that causes CADASIL? Because I really thought that if I understood the mechanism, I could find a treatment. You know, more than 25 years into it, I do have the feeling that it was worthwhile. I think I learned about the biology, but I also feel like we spend too much time. And I spend too much time thinking about the biology and the mechanism because that's not necessarily what the patients want. What the patients want are therapies. So part of this work was for me, or the result of me trying to change my mindset, not about what I want to understand as my priority, but focus on more on what patients want. And what patients want, I had a meeting with families recently, CADASIL families, and I asked them, what do you want? It's like a therapy, please. It's like, when? It's like, now. That is what patients want. So part of this, or what I want to tell you is how I focus on that. And and there are other mutations in NOSH3 that are not CADASIL mutations because they don't involve these systems, but they essentially kind of like make the protein not work or not even be there. And these type of mutations also also cause something very similar to CADASIL. And again, to talk a little bit about what we biologists and scientists spend a lot of time like talking about and researching. So when you have a system mutation in NOTCH3, it gives CADASIL. If it's not a system mutation and it just kind of like eliminates the protein altogether, it causes this disease that is essentially in the brain, cerebral. It's also autosomal dominant. It causes subcortical infarcts and it causes leukoencephalopathy, but you're not supposed to call it CADASIL. And you see here the contradiction, but it's the same thing. Yeah, maybe not, maybe one is more severe to the other one, but at the end of the day, it's really showing us that NOTCH3 variations, CADASIL type or loss of function type, they result on something in the brain that looks kind of like similar at some level. And this is going to become relevant later because the drug that I'm going to be telling you about today can help people that have these mutations, and it can also help people that has these other mutations. We have evidence indicating that it should be able to help anyone that has a mutation with NOTCH3 in NOTCH3 and has cerebral or brain pathology in their vessels. Okay. Now, here I'm going to start showing you some of this data. Again, this is a lay audience, so I don't want you to like, it's a little bit gruesome to show like pieces of brains, but it's kind of like we need to do that for research. And this is important to show and to know because sometimes we can do too many experiments in Petri dishes and in animals, and it has nothing to do with what's happening in people. So yeah, we found the link between RANX1 and CADASIL in human brain of CADASIL patients. And that's a good thing because it shows that it's important for people. Now, I want you to understand a little bit of how kind of like the brain works. We have this understanding of the brain as a neurovascular unit. This tube represents a vessel. In the vessel, we have vascular endothelial cells, and we have these other cells that are the vascular smooth muscle cells or pericytes. I'm sure you heard about them. The focus of most CADASIL research, 99% of CADASIL research, has been on these cells, the smooth muscle cells and the pericytes, kind of like the muscle around the artery. Why? Because, well, that was the most obvious thing. That's the one that degenerates, and that's where NOTCH3 is. There is some work on the vascular endothelial cells, but there is not as much focus on these other cells that are also part of the neurovascular unit, which are the astrocytes and the microglia. These are support cells, and the microglia cells are inflammatory cells that are around the vessel. And they seem to be very important, but nobody has really worked that much on these other cells. And of course, we have the neurons. The neurons are the ones that ultimately die when they lack oxygen and nutrients because the vessels burst or they collapse. Knowing this, so what we did with the tissue is that we got the human tissue from CADASIL patients after they die and also from controls, and we look at gene expression, all the genes that they have, which ones are making proteins. And in looking at that analysis, we determined that there were some processes that were affected. One is inflammation, that was new. There are other problems of the arteries of the brain that are classically known as inflammation. They are called vasculitis. Vessel is inflamed. But that wasn't really very characteristic. So we learned something new. And you can interpret this data here. Anyone can interpret it. Like white is cold and red is hot. That means a lot of expression. So as you can see, there is a lot of red on the CADASIL side for inflammation, also for angiogenesis, and also for notch signaling. And notch is important for CADASIL, but we knew that. That's not new. Angiogenesis, that was a new finding. Inflammation was a new finding. So here we discovered that these two processes, inflammation and angiogenesis, were important. Inflammation, everybody's familiar with. If you hit something or it gets infected, there is inflammation. Angiogenesis has to do with vessels, and it's this process where you can have a stable vessel or a vessel that becomes reactive and kind of like the blood and the liquid is supposed to be inside the vessel. And instead, when you have the process of angiogenesis activation, there is leakage. So you can think about it like a hose in the garden. The water is supposed to be inside, and then you put it where you want it. But in this case, when you have angiogenesis, the vessels or the hose becomes very leaky and it leaks. Not a good thing. And this is a problem, actually. And again, this is we have seen a problem in vascular permeability in CADASIL, but we did not put that in the context of angiogenesis. And this is where the two projects were like in the lab and not really, I didn't make the connection. Oh, here in CADASIL, we see vascular permeability. And here in my angiogenesis projects related to RANX1, we also see angiogenesis. Are these things connected? I didn't see that at the time. There are other characteristics of angiogenesis, which is, cell migration, proliferation, and all sorts of things. But I just want you to focus on the vascular permeability. Now, in my lab, we knew that RANX1 was very important for angiogenesis. And after getting this result, the angiogenesis was activated, we asked the obvious question. Is maybe RANX1 playing a role in angiogenesis in CADASIL? So we look at the tissue, and here I'm showing you, this is a piece of brain tissue from a control. In brown, we can stain or try to detect how much RANX1 there is, and there is not that much. As you can see, it's very light brown. Instead, you don't have to be a scientist. Everyone here can see that there's a lot of brown in the CADASIL brain. And it looks like a tube, right? So that was indicative to us that maybe there was more RANX1. And we quantified that. These high bars indicate that there is more of that in CADASIL. And it was surprisingly, yes, there was more RANX1, but surprisingly, there was more RANX1 in the microglia. Remember those cells outside the vessel that were inflammatory? So here we start to establish some connections. Oh, it's angiogenesis, RANX1, inflammation. Maybe these things are connected. So the microglia was one of the main cells that was expressing RANX1. And, we validated that in many ways. We did more expression analysis, quantification. We can look at the protein, these bands. There are ways to measure gene expression or protein expression. One is globally, the other one is these stainings. The other one is we look at bands in a gel, and the thicker, darker the band, the more expression. And you can see here how there is a lot more of this dark band in the CADASIL compared to the control. So we validated that result in one brain, then two, then three, then in a brain with one CADASIL mutation, another CADASIL mutation, another CADASIL mutation. So it was consistent across each of these bands, indicates like a different brain. So this is not a one-off. This is kind of like very consistent result in CADASIL brains with different notch3 mutations. And so the link was established, and we also asked the question, well, okay, it's in humans, but we cannot just jump to humans. And the humans that we tested were like, they had passed. So can we also find the same result in animal models? And for that, we investigated the role of RANX1 in CADASIL mouse models. So here, or we can call it like NOTCH3 mutations, because now this big distinction, is it a CADASIL mutation? Is it a loss of function mutation? It seems like at the end of the day, they cause the same thing. People get strokes, some of them can vascular dementia, okay? So here very freely, I'm gonna talk sometimes about a NOTCH3 knockout, basically NOTCH3 is not there, or a CADASIL mutation. And we studied the NOTCH3 knockouts, the ones that just don't have NOTCH3. And what we observe here, I want you to become very familiar with this image. I am at the Eye Institute. So we look at the eye of the animals or people, and we can look at the arteries. So each of these is kind of like an artery or a vein in the mice, and the mice are alive. So we perfuse with the dye, and then we can just look in the eyes of the animal, and if we see the dye, it's because it went through the vessel. So we can see the shape, the thickness of the vessel. And let me show you something here. In the wild type, that means normal mouse, we see a thin vessels, that's an artery. Like kind of like wider vessels, that's a vein, doesn't have muscle. That looks normal. Look in the NOTCH3 knockout. This one doesn't have NOTCH3. There is not such a big distinction. I cannot really tell if there are very thin or very wide vessels. They all look the same. What happens is, because the arteries lack the small muscle cell, they get wider, they get really fluffy. And here, they are so fluffy. I'm sure you heard about this, an aneurysm or microaneurysm, right? CADASIL people get that. So here we have an aneurysm, the vessel kind of like blows up like a bubble. We can rescue this phenotype. I'm not going to focus on that. And here is a CADASIL mutation. This is a mouse that has a CADASIL mutation. Remember, the C stain followed by a number followed by an R. And you can see that this phenotype of vessel abnormality, vessel leakage. Look at how here between the vessels, there is no green. That means there is no leakage. But look at what happens here in the CADASIL between the big vessels. There are like a small, there is like this signal here in between, that's leakage. So again, we can reproduce some of the pathology in this animal model. And when we do gene expression and staining, we were able to see the same thing that we found in the human brain. We were able to see an increase in RANX1 expression in the microglia. And we're able to detect that also here with the staining. Here is in green. So this is in the normal mouse, less green, NOTCH3 knockout, more green, that's RANX1. So this is important because you don't want to find something in only one system. So here we validated the same result of RANX1 increase, hyperactivation, overexpression. We can call it, there is just more RANX1. And we found that in human brain, coming from CADASIL patients, and also in mice that have a NOTCH3 mutation. And again, these are our quantifications. Higher bar means more. And that was specific in the microglia. So knowing that, we knew in the lab that RANX1 is good. RANX1, there's nothing wrong with RANX1. It's a transcription factor. We all have it. Now, RANX1 overexpression, RANX1 hyperactivation, RANX1 doing too much of what it's supposed to, that's bad. So people have developed RANX1 inhibitors, things that block this RANX1. And there is this one that is very interesting molecule. It's called Ro247429. For the purpose of this talk, I'm going to call it RORI. It's like R-O-R-I, RORI. Why RORI? Is the Ro RANX1 inhibitor. It's a RORI. We can call it RORI. It's a drug. This molecule was first discovered in the 1990s as a small molecule. Here is the shape of it, that bound a different protein. It kind of like interacted with a different protein made by the HIV virus, the one that gave people AIDS. And that protein was called TAT. And it was developed, back in the 90s, People, they didn't have good drugs for HIV like we do now. So they tried to develop this molecule. They didn't know it was a RANX1 inhibitor. They just discovered it as something that bound a part of HIV. And the drug passed all the tests. They made a phase 1, they made a phase 2 trial. 96 people received this drug in a phase 2 trial. And guess what? For those patients, for that particular interaction, RO24 didn't work. It seemed like, HIV, there are just too many copies of HIV, and they couldn't give enough of the drug for it to work. But the drug was safe. Later, better drugs for HIV came about, so the drug was dropped. Not because it wasn't safe, it's just because it wasn't good enough. It just wasn't good enough for HIV. The drug was forgotten until 2012. A very remarkable scientist, Dr. Paul Liu, he was looking for RANX1 inhibitors, and you do a screen of thousands of molecules. And he found out that RO5 and RO24, two very similar molecules, not only interacted with TAT, they also function as RANX1 inhibitors. And he published that. So that came out of the NIH. National Institutes of Health. Dr. Paul Liu reported that RO24 was a RANX1 inhibitor. So in my laboratory and in the laboratory of Dr. Leo Kim, we got excited about this. And we were like, well, this is great. There is a drug that has been in humans before. It was found to be safe. It didn't work for HIV, but maybe it works as a RANX1 inhibitor for the diseases that we're interested on. So this is called a repurposing of a drug. A drug that was made for a purpose is used for a different purpose. And why is that a good thing? Well, remember, in case anyone has any doubt, I ask people, what do you want? A therapy. When do you want it? Now. So the standard that we don't have treatments for CADASIL that kind of like really impact the pathology. There is only symptomatic treatment. So if we want a therapy and we have a candidate, the candidate will most likely have to go through the standard path. The standard path is you discover the candidate drug, then you do preclinical testing, which is kind of like all these studies in petri dishes and mice. Then you do clinical trials. Then, if you're lucky, you get approval. You need luck, you need a lot of money, and you need a lot of time. How long? Anywhere between 12 to 15 years. I'm sure you heard this number. That's what it takes. Now, when you have a repurpose drug, it's a shortcut. It's a shortcut. You may still need to do some preclinical work, but you can have an accelerated path towards the clinic and towards approval. How short? It could be a third of the time. It could be faster for other drugs if the drug was already approved for something. In this case, the drug was not approved for anything. But it made it far because you did a phase 1 and they did a phase 2. Phase 2 trial means more people. They took it for like 3 months. And another advantage of this drug is it's an oral drug. It's a pill. They took it as as an oral drug. You can take it in the comfort of your home. And that's important for CADASIL patients because they have mobility issues. I'm sure many of you, kind of like care for somebody that has mobility issues, or you have the mobility issues yourself due to the strokes, okay? So this is not like, I know people want a therapy and I know people want it now. This is not now, but it's a lot shorter than if it was the standard path. No, I don't have something like right now. But if we successfully do this repurposing effort, we can have it in a lot shorter time than if it was the standard path. So here's some, this is a paper. That trial was published. People can look at this paper. That's why I put it here. There's a reference. It tells you everything that they found in these people, how it didn't work, how it was safe. What are the advantages of focusing on RANX1 inhibition as a therapeutic approach for CADASIL? Well, this drug was used in a phase 2 clinical trial. There is a complete pharmacological review that was done way back then. It's a small molecule. Small molecules, a lot of people in the rare disease fields, they get into biologics, gene therapy, all those things have CRISPR, stem cells. Let me tell you what all those things have in common. That's expensive. How expensive? Very expensive. It just goes from expensive to very expensive. That's the range up there. Small molecules don't necessarily have to be because you can mass produce them. It's a chemical. But small molecules could be cheaper. They generally are. Cheaper than what? Cheaper than the biologics or the gene therapies or the stem cells. I'm sure you've been told about these things. So now, when we say it was found to be safe, yeah, it was found to be safe. The only side effect that was determined, and it's important for me to tell you about this, was they gave it to 96 people. They tried different doses, 75, 150, 300 milligrams per day. In 17 patients, they developed a skin rash. And in some of them, they had to stop the drug due to the rash, but it went away. So what happens with the rash? If you're going to get it, it seems that you get it soon during the treatment. And then if the drug is dose dependent, so the more of the drug, the more of a chance that you get the rash. But then if you take the drug away, the rash goes away. So as far as side effect goes, it was very mild, and the drug was considered to be safe enough for the therapy, depending on the condition. You may not want to use it if you have a skin condition, but if you have CADASIL, you get strokes, you get dementia, it could be that this safety profile will be acceptable. But again, it's important for me to tell you what they found. Now, that's again, that is an advantage of a repurposed drug. You know what to expect. Yeah, if we bring this to the clinic, hopefully not, depending on the dose we use, hopefully not. But yeah, some people may get a rash, but we knew about it. What happens in most other efforts for drug development is that you find something is new, you did not expect it, and nobody likes it. And that's why they say that drug development is, the success rate is so low. It's very low. It's because you don't know. But in this case, we know. Yeah, we know what to expect. And people can even enroll or not on the trial, depending on whether or not they accept that risk. For the other efforts, is risks unknown. Here we know the risk. This is it. I just show you. It's published. Okay. So now, thanks to the work by Dr. Kim and I, this drug is actually commercially available for research purpose only. Don't go ahead and buy this drug. I don't recommend it. When they make these things, they make them so that you can put it in petri dishes and in animals. But this is a drug that people, like there are some scientists here. You guys can go ahead, buy it, try to replicate the work that we are using in your own models. If you are a patient, if you are a relative of a patient, just look. This is not for you to take. It's not safe. And it's not safe simply because it was not made for human use. But it is commercially available. So people can buy. It's commercially available because Dr. Leo and I resurrected the drug. So now, since we published with it, now people can buy and do more experiments, and this helps. So what did we do? We took a CADASIL model. We wanted to test efficacy of the drug. And for that, we went ahead and had our CADASIL mouse model. This mouse already had pathology. They were 6 months old, and the pathology was severe in the arteries. And we treated them with the drug. In this case, we injected it, but this drug can be taken as a pill. Just for convenience, it's easier to inject a mouse than make a mouse take a pill. If you don't believe me, try. They will bite you, pretty much. Okay, so we inject, but this drug can be taken as a pill. The good thing is, in terms of our experimental design, is that mice already had disease, and we only treated them for 1 month. And what we found was remarkable. So here is again kind of like a normal mouse. You are familiar already with these images. You have the veins, the arteries, all of that. This you can tell is a CADASIL model. Everything kind of like looks fluffy. Arteries and veins cannot be distinguished. Everything is kind of like really just kind of like dilated. After 1 month of treatment with the Raw 24 with Rory, the results were obvious. This is one of those results that you quantify because you have to publish it. But I'm sure you can tell the width of the vessel has recovered. The vessels now kind of like, you can tell that there is an artery, there is a vein, there is difference here. It's not as wide as fluffy. And here is the quantification of that in the wild type. You see this range of vessel caliber. In CADASIL, they all get wide. With the treatment, you recover this variation, and in general, you lower the caliber. That means the vascular tone is recovered after treatment. I honestly, when I first got this result, we got it with the Notch 3 knockout, the mice that didn't have the knockout. Then we tried with these animals. It was really hard for me to believe because it just seemed impossible. Like I knew in these mice, they didn't have the smooth muscle cells. I was hoping to see some changes, but not something so overt. The results were validated in one mouse model, another CADASIL model, another CADASIL model, and repeated many, many times. In our hands, this result is very reproducible. But I didn't think it was possible because the only way for a vessel to have tone is if the smooth muscle cells are dead, and in CADASIL, smooth muscle cells are dead, or so we thought. As it turns out, here I'm showing you, we took those same tissues, we stained them for a marker of the smooth muscle cells here in red. This is in the wild type. In CADASIL, the red is gone. But sure enough, in CADASIL, the staining came back. And then we started to notice something. In CADASIL, the red is not here, but there is red in other places. We thought the smooth muscle cells were dead, but maybe they were just not expressing the marker or they have left the vessel. And once we inhibited RANX1, it seems like the smooth muscle cells were able to go back to the vessel and give it tone. That was a remarkable finding, and again, we quantified. Here, you see like black circles, white circles. This is because we were able to put, the result was so consistent, we were able to put together CADASIL mice with different mutations together, and it still works. We did that in the retina, the part of the eye that helps us see, but we also reproduce some of those analyses in the brain. So very remarkable result. So with this, I'm getting to the end of the presentation. I want to definitely give some space for questions, but this clearly shows that RANX1 inhibition restores vascular reactivity in CADASIL mice. How does it do it? Well, it somehow restores vascular smooth muscle cell coverage. And yes, it shows that RANX1 inhibition may be a treatment for CADASIL. A question that someone said ahead of time, like when can be this advanced? Again, I just gave you the time frame even before. We think that this, if everything goes well, it could be approved in 3 to 5 years. There are things that need to be done towards that. How can you help? Your participation here is great. Please ask any questions. There are other ways to help, which is creating awareness. You heard here and were introduced to cureCADASIL. You can help by donating to cureCADASIL. And if you have questions about the company, I'm not the person to give you that information. You can also get that information from the cureCADASIL people because they have that information and it will be appropriate for them to pass it along. With that, I actually want to end the presentation. I had a lot more slides, but they are really just kind of like a summary of all the other data that we that support this, looking at the eye diseases, looking at the lung diseases. You know, that long list of papers that I showed you at the beginning, like I had all that data, but again, you came here because you wanted to know about CADASIL, and we discussed that. Now, what are the pros and cons of this new therapy? Well, the pro is we have evidence that it works. How well it works. In these models, it works really well. Another pro is that it seems to work on some of the medications or therapies, experimental therapies that you might have heard about. They could help like people with this mutation, but not that mutation. They are too specific. This one seems like it could help people with any NOTCH3 mutation and any CADASIL disease or something that looks like it. Another big pro is that the drug has been tested in the clinic, and we know it's safe, and if there is a risk, we know exactly what the risk is. Also, something big is the isolate administration. I worked before on an antibody that's a biologic. Remember the expensive ones that you need to go and get infusions every 2 weeks? That might still work, but it's cumbersome for CADASIL. Cons. Well, I think I posted about that, talking about repurposing drugs. Everybody loves to talk about repurposing drugs. Oh, if we had a drug that had been in the clinic before and it worked, wouldn't that be great? We can bring it to the clinic and save us all. Guess what? Nobody wants to invest in that. Very hard. So these repurposing efforts really need a community to rally behind the effort for it to happen. If not, people will be like, you can present this to people. Oh, yeah, this is a great idea. We can help people with CADASIL. There is no treatment. And oh, that's great. Why don't we start all over? Can you find a new inhibitor? We make it new. If it's new, you go back to the standard path. So there you go, 10 to 15 years. Why do they want to do it like that? Yeah, because, investors can make more money that way. That's just the reality of the situation. And maybe they start talking about the rashes or, whatever. But, at the end of the day, for CADASIL, and I want to end with this, yes, I've been working on CADASIL for quite some time, and I feel like I'm late. I want to help people with CADASIL, and I'm very excited about this new drug. I want to be part of bringing this to the clinic, and I need the support of everyone that heard this to make it happen. And with that, I end, and I take questions if there are any.
Thank you,
Joe, thank you very much. This has been a great, cogent review, and we've been over here fielding a number of questions. The ones that were easy, we've been already sending answers because there have been a lot of questions. But let me ask a couple of these, and Jane and Benita, you're welcome to weigh in too. But more than one people have asked this question from Katia Gautier. How can we get into the clinical treatment? I think they're asking, what is the gateway for patients to get into a clinical trial like you're describing?
Okay, so the best way to do that is the trial is not ongoing yet. There are things that need to be done before. One of them is that there are steps, like we know a lot of information, but it seems like the FDA wants us to repeat the toxicology just because it was done like a long time ago. So that needs to be repeated, and the drug needs to be manufactured. Remember, I just showed you this can be purchased online. You shouldn't do it because it's not made for human. It's not for human use. So now we have to go in and synthesize, make the drug for human use. That is kind of like a process to make sure it's safe, to make sure there are no contaminants in it. So we need to make the drug, and maybe we need to repeat the toxicology package, whether or not we have to do that specifically for CADASIL. Now, what can you do now? In the United States and in other countries, there are clinical programs ongoing. One of them out of NIH, another organized by Dr. Jane Paulson, another, another one is organized by Dr. Fanny Elahi. And these clinical programs that are enrolling patients, they are just enrolling patients. It's called clinical trial readiness. What is clinical trial readiness? Is whenever you have that freaking drug that you are late for, Joe, we're ready. So I strongly encourage you to join those programs if it's within your means and you qualify, because they are studying biomarkers, they do the MRIs. And yeah, we've been in touch with those groups so that they know about our efforts. And if and when we're ready, we can enroll patients, but the patients will be enrolled from that pool of patients, if you will. So something that you can do now is ask questions. cureCADASIL will be a good resource as to guide you toward which effort is closer to you or which one you may qualify to. I'm in the lab. I'm not a good resource. I know of those efforts, but I'm not directly involved with any of them. But something that you can do is get involved with those efforts, because the patients most likely will come for the trials, will come from those clinical readiness efforts. And again, I just mentioned the ones I know of: Dr. Jane Paulson, the Fanny Elahi, and the NIH effort as well. If I forget one, you can get the information from CADASIL.
And they are on our cureCADASIL's website,
Okay.
the 3 studies. But kind of along that line, is that, are you still collecting postmortem brains? There's a couple questions that came about that. I think people are interested in learning how they might be able to donate.
Thank you. The same teams might be able to collect the postmortem brain. I am only able to do that via my collaboration in people in Colombia. I don't have the means to actually collect that myself in my laboratory, although Mass General has a brain bank. So the brain bank will collect brains from any condition, any neurological condition. And if you look, brain bank near me, universities usually have them, and they are usually in networks, and they make tissue available. So by making a brain donation to a brain bank, it's very likely that CADASIL researchers can then go in and access the tissue, even if they didn't collect it themselves. For instance, many groups here in the United States or in Europe access the tissue from Colombia, because it's made for research. It's not like, oh, it's like it was donated to me, so it's only for my use. No, that's not how these donations work. People want to make that tissue available to anyone that needs it for research purposes. And there are some ethical considerations and permits that you need to get. But usually, if you donate to a brain bank, rest assured, other researchers, even my laboratory, will probably be able to access it. Good question.
Yeah.
I can add, if someone wants to do this, they can contact cureCADASIL, and we will put them in touch with appropriate people who can help arrange it.
Bert, can I interject with a question from Dr. Michael Geschwind? He asks, run X1 is a very common transcription factor, yes? So what happens when you inhibit such a common transcription factor to other important genes?
That is a great question. Thanks, Dr. Ganshwer. So RANX1, yes, is very important transcription factor. And when we call the process, when we were saying or promoting the idea of inhibition, is really normalization. We want to bring to the attention that for instance, in the brain and in the retina, we haven't seen a lot of normal RANX1 function. Is when there is pathology that lights up. And the small molecules, the way they work, what they do is that they inhibit, they don't block completely RANX1 function. Actually, we have some data. I'm not promoting here like doing a knockout of RANX1 or complete abrogation of RANX1, no. But I think there is what we call a therapeutic window, in which by using the proper dose of a RANX1 inhibitor, you can have the beneficial effects without totally knocking out the function of the gene. So therapeutic dose is appropriate. The advantage here is that I think the therapeutic dose will be probably in the lower end of what was used in the HIV trial. RANX1 is very sensitive to this inhibitor, and we don't feel like we need to do kind of like very frequent treatment. I can tell you the mice, we treated them for a month, but we only treated them every other day. Like every other day we gave the treatment and it still worked. For a small molecule, that is substantial, given it every other day. Usually, a small molecule, you're like, what's a small molecule? It's like, I don't know, I was going to say, is that a good thing? The good example used to be Tylenol, but that one is kind of like in the doghouse. And aspirin, and those are, Advil. Advil is good. Let's go with Advil. If you have a kid, you have ever taken Advil, you take it 2 or 3 times a day. Okay? So that you take it just by mouth, So small molecules, usually you take them off, and we use them every other day, and it still works. So I think we would need low dose, and the therapeutic goal is normalization, not total blockage. I wouldn't promote, for instance, doing a knockout. Others do CRISPR, get rid of RANX1. No, but the small molecule could work. And again, in the models that we have studied and in the human trial, it seems like these doses were not associated with severe pathology or side effects, and it was probably because you still had some of the normal RANX1 function. That's a great question.
I have a couple more that are related.
Yeah.
So Joe, there's question, and what about the people most affected? Could this reverse their sequelae? And another related one, if vascular reactivity was restored in the mice, did their CADASIL symptoms, apathy or dementia, improve? I don't know if you've tested symptoms in mice, but nevertheless.
Okay, so that is the part of this study that I think is reassuring. The mice already had established pathology, and despite having established pathology, we were able to restore the vasculature. So yes, I think this therapy could probably be used preventative for people that don't have the disease but they have the mutation. But probably it could also help people that already have the disease, because I just showed you that the mice that I use already had very severe pathology in the vessels, and yet they improve. So yes, now mice, it's not that we can really look at apathy or that these mice get strokes, but like they have severe pathology, but not that severe. What I would say is, would it help with the stroke recovery? I don't know, maybe not, but hopefully it might reduce the chance that you get another one, something like that. That I consider more viable. But I want you to know, for instance, something very practical. When you go and get the MRI and you see these white spots, that is promoted by the vascular leakage. So I will anticipate that with this drug, by reestablishing the vessels and the vascular tone, we'll be able to see a reduction of these white spots in the MRI. The same in the retina. We will see a reduction. I mean, I show you that. For the MRI, I didn't show you that. I don't have that data. But for the retina, we show that. But we cannot really look at very advanced or human-like. For that, we'll need a trial. In the trial, we'll see if it helps with apathy or if it helps with like getting fewer strokes. That could be an endpoint. How many strokes per year were in the control versus the treatment? Did it get reduced or it didn't? Because that will show us if it's really working. This is what we call an experimental therapy. It is still experimental. We have to do the trial to know if it works. I don't know if it's going to work in humans. It works in mice. It works in these models. It works really well. I think it's promising. Anyone that tells you, oh, this drug will work in a human, I don't know, they're selling something. It's not, the only way to know if something works in a human is to do a clinical trial, okay? That's the only way. So we have to do the trial.
Okay.
I can slip in a quick question here. Someone's asking about the half-life of the pill, and wondering, how often it would have to be taken in a clinical setting.
It depends on formulation. Like I said, our formulation, we have done it every other day. I will anticipate that depending on how we formulate the drug, it may need to be taken 2 or 3 times a day, but formulation can be improved. Ideally, it could be done once a day. Let's see where we get. I'm not, to be honest, I'm not that worried about formulation right now. I just want to know if it works. If it works, I think, but it has to be somewhat convenient anyway. But I would say anywhere between 1 to 3 times a day would need to be taken. But hopefully it will be one of these trials where you can give them pills to the patient and they go and come back like 2 weeks later for the next test or a month later, So hopefully it will be like that. But we need to work on formulation. I've been worried with formulation, but sometimes out of convenience, or we know this works really well for like the eye. Like I say, in diabetic patients, they have like problems in the arteries. So we have a eye drop. That one works. I can tell you, and we did the formulation in my lab. That was done by Dr. Said Arevalo and David. So we have eye drops for that. That works really well. We did it in rabbits and we did it in mice. So I like, but again, I'm a scientist, so we like to these trinkets.
I have a related question there, though. Someone is asking if there's value of looking at the retinas of individuals with CADASIL.
Absolutely, and I believe one of the efforts by Dr. Genschwer and Jane Gunter, they included the retina studies in CADASIL patients. That is ongoing. Actually, I will encourage people to participate on those studies. Again, because all these things that are happening led by other people, they enable the future trials. So if they manage to validate the retina imaging, the eye imaging, then we can use that for the trial. So yes, big time. I mean, it works. I just show you. It's a great endpoint, and there are differences in CADASIL patients. Yeah, for sure.
That retinal work is being done in Dr. Jane Paulson's CADASIL Consortium patient history study. I don't think this one has been asked yet. This comes from Professor Dr. Raj Kalaria. He said, "Thank you, Joe. Great talk. Do you think it will be more effective in those with non-cysteine mutations?"
Hi, Raj. It's great. Thanks for the question and thanks for being here. I miss you. I have evidence that it works for both. So we tested in animals with the C455R mutation. We tested in mice with the C1031 to R1031 to C mutation, and we tested in the NOTCH3 knockouts. It works in all of them. So, and again, why does it work in all of them? Because it's kind of like mostly impacting the microglia. So it reduces this inflammatory proangiogenic factor coming from the microglia. So I think it will have an impact in all of them because it's not related to like cysteine interaction, or it's not related to like NOTCH3 accumulation. Is because it's indirect, it may help people with different mutations. Having said that, and one of the purposes of this talk is to tell everybody that wants to hear it, show them where they can buy the drug and welcome them to try in their own models. We have a paper specific for the CADASIL work, is under review, and it got peer reviewed already. And we went through like 133 comments from the reviewers, and hopefully will be published soon. But part of the reason for this presentation is to encourage everybody to try in their own models. Try cells, try the mouse model with your favorite mutation, and tell me how it works. Publish it. Beat me if you can. It's just kind of like you cannot. The idea is that the patients win. Kind of like we need that type of study, but I only have access to some mutations, not all of them. So people should do these studies. I encourage it because it seems reproducible. So I don't think you're wasting your time or your efforts if you go in this direction. Stem cell, organoids, try. RANX1, I mean, ask Dr. Leo Kim, and we are invictus. This drug works. It works in the eye, surface of the eye, the retina. It works in the lung. I didn't show you that data. It works in many tissues. It works for fibrosis. It works for angiogenesis. It's a good mechanism. I think, RANX1 targeting, and this goes back to Dr. Genschwer. RANX1 targeting will be in the clinic and will be effective in the clinic. For which indication is going to be approved first? I don't know. I prefer if it's CADASIL. I have my favorite, my favorite disease to target will be CADASIL, but RANX1 targeting will be approved in the clinic for some indication in the near future, considering everything that is happening in the field. That's my take.
Joe, there's a question here from Dr. Fabrice Dabertran. Said, thank you for the great presentation, impressive results. How do you see the RUNX1 pathology model working with late onset of CADASIL? One could expect to see the symptoms appearing at younger age.
That's the thing with CADASIL, it is progressive. So I don't know if people with advanced disease, we can recover what was lost, but hopefully we can stop the rate of progression. Because now we're talking like people that have had a lot of strokes, already has dementia, but not getting that other stroke, which sometimes is, my experience with patients with CADASIL, the fear is the next one. It's kind of like, I already got one, okay, I recovered, I made it, but it's like the fear of the next one. So if we can reduce the rate of that, or like they don't progress in cognitive decline, that will be, I'm glad that people are asking this, because I will consider that a win. I don't want to, there is a difference between, I'm a very optimistic guy. I'm very excited about this one, but we also have to kind of like what we have evidence for is, and I think is achievable, is stopping the progression. The idea of recovery of what was lost, I honestly don't have evidence that I can do that with this drug, right? Because that's kind of like different. That's kind of like restoring neurons that die and things like that. We can show that vessels get invested again by small muscle cells. I show you that. Vascular tone is recovered. I think that what I will expect is vascular tone recovery. I will expect reduction in micro hemorrhages and microinfarcts, reduction in rate of or the size of leukoencephalopathy, all of that. I'm hopefully stopping the progression of the disease. No, I don't think I have any evidence that supports the idea that you lost something and that will be recovered. I mean, that is something to hope for, but I honestly just don't have evidence for that, and I have to be careful on that. But again, for a condition for which we have nothing, I consider that a win. I'll take it as a win. I don't know. You guys can be disappointed. It's fine. It is your job to be disappointed. You guys have to push us, demand more, but I'll call it a win.
Joe, there's a question that I know you've considered and are working toward, but someone has asked, have you thought of FDA fast track?
It will be ideal. It will be ideal for that. Again, that's that's something that, maybe the company has to drive. I think there is an argument for that. There is something I want to do, which is I want to get orphan drug designation. That I want to do. I'll do that. It's kind of like that accelerates the process, but that's within my means of being able to do, and I don't need any company to do that. I wanna get an orphan drug designation for Rory. I'll do that, and that accelerates the process. Everything else just really has to do with what I can do from my academic post, and what a company will do, or a partnership with cureCADASIL or other institutions if they want to engage to a fast track. I cannot do this alone. That is for sure. Or even again, for rare diseases, if you look at how people with rare diseases get things done, they don't delegate. You have to, pick up you have to do it yourself. You have to get involved, and these people are taking too long. I'm just going to go ahead and do it. What is it that is done? We will push the FDA. We will get engaged. We will advocate. We will make noise. We will do it. And it's up to you guys. To be honest, that's part of the reason to talk about the discovery and try to engage the community, is to empower you to do things, because I can only do so much. It's just and the time it takes for this whole thing to happen. It's a good one though, compared to other things I have done. Yes, I work on an antibody. People know about that for NOSH3. I work on gene replacement. Yeah. I have worked on this for actually 27 years since I started in med school. This is the best one I got, hands down. I think it's repurposed, can get us fast. It's a small molecule. I can see patients taking this. I don't want to have my patients having to go every 2 or 3 weeks to get an infusion. Mobility is a big deal. This can be cheaper than a biologic. It's the best one I got, and I think it checks a lot of marks. I would say has been already in patients. I don't know. It checks a lot of marks. You guys can do your own checklist, what you're gonna support or not. But for me, it checks a lot of boxes compared to, and it's also broad spectrum. We're gonna do something for one specific type of mutation versus another one. How much resources go on this? This one checks a lot of boxes for me, to be honest.
If there are no other burning questions, Jane and Benita, I think we'll call it a day here, Joe. And let me
Yeah, thank you to cureCADASIL, Bert, Benita, Jane. Thanks for organizing this, for inviting me, all the attendees. Thank you so much. I really appreciate the time, and thanks to my collaborators and that helped me make these discoveries. I really appreciate it.
Yeah, no, we thank you. It's been great, very informative, and I think a lot of questions were answered here today, and we're really looking forward to this progressing, ultimately coming to clinical trials and therapy. If you can see my last slide up here, there were a couple of questions that people were asking. This webinar recording will be available very soon. And you can visit our website. We will definitely post a link there. So if you want to go back or have others look at this, I think that would be very well. So again, I think on that note, we will call it a day, and I hope all of you join us in our next webinar. Thank you.
Thank
Bye.
you, Joe.
Well, thank you, guys.
Yeah, thank you very much. That was terrific. That was terrific. You've generated a lot of excitement.
Well, I really appreciate the opportunity. I am excited, so it was kind of hard to hide, and it's contagious, I guess.
Yeah.
I have to run now, but thank you so much.
Okay. Take care.
Thank you.